All right, everyone, welcome to Grand Rounds. So before I get started introducing today's two speakers, I just want to highlight that next week we're actually going to have Tate Shanafelt visiting from the Mayo Clinic to talk about organizational efforts to cultivate clinician well-being. It should be a great talk. He's coming to do some other things on campus as well. So please attend and share with others. But today we are really lucky to have two of our own faculty as speakers to talk a little bit about how they have collaborated in team science within their division. So first we have Dr. Helena Jonsson, who received her MD PhD in immunology from Washington University in St. Louis before doing her internal medicine residency and rheumatology fellowship at Brigham and Women's Hospital. She is currently an assistant professor here in our division of rheumatology. And her research focuses on the immunopathogenesis of rheumatoid arthritis, with a particular emphasis on tissue-resident CD8 T cells and granzyme K-mediated effector mechanisms. She currently leads multiple NIH-funded projects and an R01 examining the role of granzyme K expressing CD8 T cells across autoimmune and inflammatory diseases. She's received numerous awards, including our Rising Star Award here at the University of Colorado. And she has published in many leading journals, including Nature, Nature Immunology, Science Translational Medicine. And then we also have Dr. Fan Zhang, who earned her PhD in computational biology from Worcester Polytechnic Institute, and then completed postdoctoral training at the Broad Institute of MIT, Harvard, and Brigham and Women's Hospital. She serves as an assistant professor in the Division of Rheumatology with a secondary appointment in biomedical informatics. Her work focuses on advanced computational and AI-based methods to study immune-mediated inflammatory diseases using large-scale single-cell and spatial multiomics, and is the principal investigator, again, on multiple NIH-funded projects. She has received many prestigious honors nationally and here with our Rising Star Award as well, and has published in New England Journal of Medicine, Nature, Nature Immunology, and the list goes on. So I'm really excited to have both of them here to talk about how their work has intersected, and welcome them for Grand Rounds today.
Thank you so much. All right, thank you so much. This is wonderful to have this opportunity to share our projects, both individual and collaborative projects with you today. So Fan and I are going to be going back and forth a little bit, but I thought I would start, since this is the medicine Grand Rounds, and start out with a little bit of a clinical case. But all of this is going to be surrounding the topic of bite to bedside, really integrating computational to bench research, to clinical trials, to clinical progress, predicting progression and new targets in rheumatoid arthritis. So a clinical case, and this is pretty heavily based on someone that I met just a couple of years ago in the rheumatology clinic here at the University of Colorado, but is also representative of many, many patients that I've met over the last 15 years or so that I've been doing rheumatology clinical care. So a 35-year-old woman, she recently gave birth to her second child about 2 or 3 months ago, and she presents with swelling, pain, and stiffness in her fingers and her wrists and her feet, a little bit in her knees and other joints as well, potentially. Her mother has rheumatoid arthritis and her sister has lupus, and so she kind of knows what's going on when these symptoms start. This isn't terribly new to her. We do some blood tests and they show a rheumatoid factor of 215 and anti-CCP antibodies of 160. And so those are both quite elevated, and the diagnosis here is easy to make. This is seropositive rheumatoid arthritis. And so these are some of the questions that many of these patients ask me when they meet me in clinic. One, especially for someone like this, when her mother has rheumatoid arthritis, her sister has lupus, like, can this be prevented? She's also thinking about her children. She has 2 children. She already has older relatives who have this. What are the risks for her children? How can they avoid getting rheumatoid arthritis and lupus like her relatives? What happens now? Like she has this diagnosis, like what does this mean? Is she gonna have this pain forever? Is she gonna feel better? And if so, how long will it take? And will my hands end up looking like this? Because if her mother was diagnosed in, let's say, the 70s, her hands may very well look like this because it's only been in the last 20 years or so that we've had treatments that are effective. And so she's also terrified not just of the next 6 months of being able to pick up her newborn without being in pain, but also about, like in 20 years, will I be able to hold a fork? Like, will I be able to open a jar? Will I be able to empty my own pot of spaghetti? So the answers to those questions, could this have been prevented? No, at least not yet. And this is something that Fan's going to be talking about, because this is something that the University of Colorado is actually like world leading in trying to figure out how to prevent rheumatoid arthritis from happening. So stay tuned for just a few minutes on that. What happens now? As a clinician, that's actually a very easy question. Unless she has a contraindication, what we're doing next is starting a medication called methotrexate. And that's been the medication that we start for rheumatoid arthritis since approximately like 1985 or something like that. People are probably a little disappointed now that in 2026, we're still using something from the 80s, but it's tried and true. Unfortunately, we do not still, after using this medication for 40-something years, have any way to determine if this person's going to respond to it or not. It's a total coin flip. Every single medication we have in rheumatology clinic, it's about a 50-50 chance that people will respond or not respond. We have no blood test to predict it. We have no genetic test to predict it. We can't even do synovial tissue testing. Like there's no fancy test that predicts who will respond. And so she might feel better in a couple months, or she might not. Sometimes it takes us 2 years to work through all the different medication options before we're finding something that works. I feel like we can do better, hopefully soon. When can I expect to feel better? Again, hopefully in 2 or 3 months because of the methotrexate. But maybe not, and if so, then we'll add something else, and if that doesn't work, we'll switch again, and we'll switch again, and we'll switch again every 2 or 3 months until we find it, and hopefully it will be less than 6 months until you feel better, but it might be 2 years. And unfortunately, if it takes us 2 years to get your disease under control, we're unlikely to ever get it fully under control. Whereas if we can get your disease under full control within 3 to 6 months, your prognosis in the long term is much better. So if we had a test, we would not only save people pain, we would also save people from getting hands like that. We would save them from having knee replacements later on. We would save them from a whole bunch of different things if only we had a test. And so if you don't believe me about the coin flip, here's a graphic. This is from 2018. We cannot make one that looks any better now because we've made no progress. So in rheumatoid arthritis, we have many different kinds of medications to use. Here you see co-stimulation inhibitors. So that's abatacept, brand name Orencia. It's essentially the opposite of checkpoint inhibitors used to treat cancer. It has about, so here what's listed, let's see, I think this should be a laser pointer. Here we go. So apologies to those on Zoom who can't see this. But this is listing the percent of people who achieve a 70% response. So let's say this woman has 20 inflamed joints, like about 10 or 15 on her fingers, because each finger has 3 joints. So it's very easy to reach, about 8 or 10 there. And then her knees and her ankles. 70% improvement means that you still have a lot of pain in 6 of those joints. Imagine going through every day of your life with pain and stiffness in 6 joints. But that's our goal. And for this medication, even coupling it with methotrexate, we max out at 32% having a 70% improvement. For TNF inhibitors, only 24% have that level of improvement. For IL-6 inhibitors, IL-6 receptor inhibitors like tocilizumab or sirolimus, 38%. Rituximab, a B-cell depleter, that's our highest one here at 47%. And so you can see I'm actually being generous when I say 50-50 coin flip, because actually it's a little bit lower than that. Okay, so hasn't anyone tried to figure this out? Why aren't rheumatoid arthritis patients all the same? Why don't all rheumatoid arthritis patients respond to all medications, right? If people have the 2 blood tests, wouldn't you think that they would respond? Kind of like if you have thyroid disease, you get thyroid hormone and that makes you feel better. And shouldn't this be easy? But it turns out that even if you have the same blood tests, what's actually happening in your joints is very different. And this is something we've known for a while. In this group, Kaz Pitsalis, he's at University College London, is probably the group that's been doing like the most work on this, trying to define the heterogeneity of rheumatoid arthritis. And so what they've been doing is taking synovial tissue biopsies. In the UK, they're really good at this. Almost everyone, it feels like, who gets a new diagnosis of rheumatoid arthritis gets a synovial biopsy. So they take a little piece of the synovial tissue from the wrist or the knee or the finger even, and they do studies on them to see like, what does that rheumatoid arthritis look like? These biopsies are rare in the United States, but are growing. And thanks to Larry Moreland, who's here today, and others, like we are performing them here at CU. But what Kaz and others have shown is that there's actually a large range of appearances of rheumatoid arthritis. And let's see if I can get this. Here we go. All right, so now everyone can see. And so you can sort of, in general terms, classify rheumatoid arthritis into 3 categories. Here I'm calling them B and T cell rich, myeloid cell rich, and leukocyte poor. And so again, these are pieces of synovial tissue, so the joint tissue that is inflamed in rheumatoid arthritis. And what Kaz Pitsalis and his colleagues do is stain them with antibodies that bind to CD3, which is a marker for T cells, CD20, a marker for B cells, CD68, a marker for myeloid cells, CD38, which is expressed on plasma cells, those antibody-producing B cells. And then we have CD79A, which is a marker for B cells that do not release antibodies, so non-plasma B cells. And you can see that some people with rheumatoid arthritis, they have a full house. They have all these things. They have B cells, they have T cells, plasma cells, myeloid cells, like everything. But you also have some people who don't have a lot of T or B cells, but they have a lot of myeloid cells. So myeloid cells meaning macrophages and monocytes. And then you have some people who are leukocyte poor, where they're essentially not staining for anything, like a little bit of myeloid cells, but mostly it's fibroblasts and endothelial cells, and not a lot of immune cells. And all of these people have hot, swollen, painful joints. All of these people will have bone erosions and joint destruction, but the tissue looks different. And so knowing that, maybe it's not surprising that their responses to medications are different. The funny thing is, when people first realize this, they're like, okay, well, this is it then. Like, look, if you have B cells in your synovium, like these folks, and then you give them rituximab, 100% are going to respond, right? No, it's still 50%. And even people here who have no B cells in their biopsy, some of them still respond to rituximab. And so even categorizing it like this didn't actually help us that much further. And so this has never become a clinical test because it's not good enough for predicting treatment response. Okay, so if these pathotypes don't predict treatment responses very well, what do we do? Well, we need a more sophisticated dive into RA heterogeneity. Just staining for T cells and B cells and stuff is clearly not enough. Like, we need to do more. Like, why haven't we done more? Well, when I tell you that people get synovial biopsies, I didn't really tell you how tiny they were. And so this is a photo, this is a photo from the actual study that Fan and I have collaborated on that I'm going to tell you a little bit more about in a second. This is what's called a cell strainer. So this thing is about the size of a quarter in diameter. Like, this area down here is about the size of a quarter. These are the tissue fragments. They're kind of about the size, if you took a ballpoint pen and then made a dot on paper, that's what each of these is. And you usually get maybe 12 or so, sometimes only 6 or 8 from each patient. That's not a lot of tissue. It's not a lot of cells. On average, in this study that we were a part of, and this is only about 5 years ago now, the median number of cells we got out from each patient was only 33,000, which, if you've done bench science before, you know that that is not a lot. And so if it takes you tons of effort and time and pain for the patient to have this biopsy, although I will note most patients who have a synovial biopsy say they would be willing to do it again because it's not that terrible and everyone wants to figure out what's going on here. But you have to have a really good technology to make these tiny biopsies worth it, right? How do we get the most information possible out of this? Thankfully, the omics revolution in science has really helped this field in rheumatoid arthritis. And I would say all of human immunology, all of translational research, because now there are a number of different technologies that allow us to get a lot of information, even from just 33,000 cells. We can do CyTOF, single-cell RNA sequencing, and then all the other variations thereof, like CytSeq, which allows you to do antibodies on the surface of cells, ATAC-seq, which allows you to look at the chromatin. There's spatial transcriptomics, which we'll talk about later in the presentation. There's CRISPR, so now you can like grow out these cells a little bit and like, delete genes and things. There are humanized mice, where you can put human cells into mice and then study them there. All these like really fancy things. The tricky part is that they're all really, really expensive, like really expensive. Like a single run of single-cell RNA sequencing costs on the order of $5,000. And if you know human beings, just looking at all of you in this room, you all look different. We have a lot of variability. To get the necessary numbers of patients to see anything, usually in clinical trials, that number is like in the hundreds, right? So hundreds times $5,000 per patient, that's a lot. And so how are we going to make this worthwhile? And the answer there is team science. And so I just wanted to introduce like one of the big team science initiatives in rheumatology that I think has been really successful and that Fan and I have been a part of, also Larry Moreland, who's here, and Mike Hollers, who's the division chief prior to Christy Kuhn, who's here. Like all of these people and a whole bunch of rheumatoid arthritis researchers across the US and in the UK all got together to do this one big collaborative project with big ambitions and a big budget. This was funded by the NIH and the Foundation for the NIH. The Foundation for the NIH is pretty nifty because it takes pharma funding and sort of couples it together into large grants that are then shared by multiple research groups to do big, big projects. So this, and don't quote me on this, but this was like roughly a $42 million project. Way, way bigger than a single R01. And that's key because if you don't have that kind of budget with that kind of project, you won't have enough numbers to be able to tell anything. So again, you remember how tiny those little synovial tissue biopsies were. And if you don't remember, I put the little image up here again. So you can remember tiny, 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 tiny samples. We wanted to squeeze every single ounce of information from these cells. And so this is the pipeline that we designed. And I won't go through it in detail. I encourage you to read the paper. The reference is on the next slide here. But we took them, like some of them went to histology. The rest were digested so that they were a solution of cells. Some of them went to single-cell RNA-seq, others to flow cytometry, others were frozen down for other studies in the future. Like we did not throw a single cell in the trash without getting information from it. And this is what it yielded. So this paper in Nature came out a couple of years ago now. If you like rheumatoid arthritis, or you like team science, or you like single-cell omics, I encourage you to read it. But this was a large study, the largest by far in rheumatoid arthritis and in rheumatology in general at the time it was published, with 300,000 cells from about 80 individuals. We had originally planned to write a different kind of paper about this, about how people who are on methotrexate but don't have a clinical response are different from people who are on a TNF inhibitor and don't have a clinical response. But it turns out that's not where the story was. The story was about how rheumatoid arthritis comes in many different flavors. And instead of just using those T-cell antibodies and B-cell antibodies, we used transcriptomics, meaning looking at the whole genome all at once to classify rheumatoid arthritis into different categories. And you can see them here. And again, I'm sorry I don't have time to go into this in detail. And so this all seems a little overwhelming. Don't worry. It was for Fan and I as well for the several months we spent writing this manuscript. And so here you can see we found that there are actually 6 categories of rheumatoid arthritis. We named them by letters for the dominant cell types. So for example, there's one category called F, where most of the cells are fibroblasts. And so the way that this graph here works is that each column, and actually I have this in a bigger version here, each column is a patient. And so we had 11 people who have this F phenotype of rheumatoid arthritis, where most of the cells are fibroblasts, seen here in light blue. There were other people who had a mixture of T cells, shown here in lilac, and then fibroblasts, shown here in light blue. And other people that had mostly T cells, but then also a lot of B cells, shown here in this sort of brick red-brown color. And so we use the sort of more advanced kind of cell discrimination to categorize these cells into 6 different categories. And it turns out that if you classify them by their cytokine production, you get the same 6 categories. Like you can classify them in many different ways, and you wind up with these same 6 categories. One thing we were lucky to do in this study is to use some of Kaz Pitsalis's data. His group had gotten biopsies from people before and after starting a treatment. So 4 months apart, they had biopsies from the same individuals. So we got a chance to see, like, if you start out in one category, let's say this like TB category, where you have lots and lots of T cells and B cells, and you receive treatment, do you always stay as CTAP-TB or do you change? Because that makes a big difference, right? Like, what if we only had to get one biopsy when people were diagnosed and say, you have rheumatoid arthritis, that's TB type, T cells and B cells, and that's the only thing we ever have to find out about you. That turns out not to be the case, unfortunately. It turns out that people can change from one kind of rheumatoid arthritis to another. It can happen over time. It can happen with treatment. It does tend a little bit to correlate with response to treatment, but some people do not clinically respond to treatment. They feel just as crappy at the 4-month time point as they did at the beginning, but their rheumatoid arthritis has still changed. And so we have a lot that we still have to learn about this. And so that's one way that this team science is being used is, let's correlate this with clinical response. Let's use all the clinical metadata that we gathered from these people and try to figure out the keys to figuring out like what treatment's going to work for who. The other way that this team science has been done is that it gave us a much higher resolution view of rheumatoid arthritis than we'd ever seen before. And so here I'm showing you UMaps of the single cell data. So UMaps are a way of showing each individual cell in this data set, and they're graphed by sort of the kind of expression that they have in their genes. So this right here are the T cells. And so there are T cells that have this, like that sort of map together over here in this kind of population, and some that are over here, some that are over here. The cells that are similar to each other are clustering together. And then based on what we've learned at the bench over these, several decades now, we've annotated them with the type of T cell that they are. And it turns out, we put this is actually like browsable on a website. So if you're really into single cell RNA-seq, like you should screenshot this because it's a super fun website. But this also led us to see and to find populations that had not been studied in rheumatoid arthritis before. Because if only 30 or 50% of people respond to any given treatment, some people don't respond to anything. Like, we need totally new treatments. And one of the ways to go about doing that is to study the cells that haven't been studied in rheumatoid arthritis before. And that's one of the things that my lab does. And so we study this population here, the ones that were labeled 5, 13, and 14, these really attention-grabbing names. But these cells, it turns out that they express a molecule called granzyme K. Now, I did my entire PhD on natural killer cells. I knew a lot about CD8 T cells. I had never heard of granzyme K before the initial version of this data set came out. But I knew that no one else really knew much about it in CD8 T cells either. And so this was something new to study in rheumatoid arthritis, a new lead that could potentially lead to treatments. And so that's what we did. And so first we validated this by looking at the protein level. So this is flow cytometry, where CD8 T cells are stained for granzyme K on the x-axis, and granzyme B, the typical cytotoxic molecule that you might have heard about in your immunology classes way back when. And what we see that in blood, we don't see a lot of these cells that express granzyme K either alone here or together. But when you look at synovial fluid and synovial tissue, you see this gigantic population up here that expresses both granzyme K and granzyme B that's different from blood. At this point, we didn't know if this was only in rheumatoid arthritis or if this was something that was seen across diseases or across tissues or across something or another. And so this was Fan's and my first collaboration as individual scientists separate from this team science project, where we got together and identified several single-cell RNA sequencing data sets from autoimmune diseases. So Crohn's disease, ulcerative colitis, lupus nephritis. This was, around like 2019, 2020 or so, where these data sets were starting to come out at larger volumes. And what we did is just looked for the percentage of CD8 T cells in those data sets that express granzyme K, shown here in these gold bars, compared to the ones that express granzyme B, this more typical cytotoxic T lymphocyte molecule, and that's shown in blue. And what you see here is that across tissues and across diseases, in the tissues, the granzyme K CD8 T cells dominate. So in rheumatoid arthritis, we'd already seen this, right? So the yellow bar, much higher than the blue. Turns out that's also true in synovial tissue from osteoarthritis. Here we have the yellow, here we have the blue. It's also true in Crohn's disease. So here we have inflamed Crohn's disease with many more granzyme K cells than granzyme B. Interestingly, it's also true in non-inflamed Crohn's disease and even in healthy colon over here, as well as in ulcerative colitis. In healthy kidney, you also see more granzyme K than granzyme B. Interestingly, and this is still true to this day, the only tissue in disease where we found equal numbers of granzyme K and B is lupus nephritis, so kidney disease in people with SLE. And we don't understand why that's the case. Even in COVID-19, if you look at the bronchoalveolar lavage fluid from people with severe COVID-19, you see more granzyme K than granzyme B. So we had a thought even here, and in 2021 or so, that granzyme K cells were actually much more common than initially realized. Since that time, it's really exploded, and granzyme K is everywhere. So Keely Mickens, my postdoc, and I are writing this review on CD8 T cells and tissues, and we're having a hard time keeping up with all these publications. Here, for example, is one where they report that having a high percentage of granzyme K CD8 T cells in acute myeloid leukemia is correlating with better outcomes. So this is the survival curve. High granzyme K is in red, low granzyme K is in blue. I have no idea what the mechanism is of this, and it's especially confusing because the very next year, someone published the opposite. So here, people who have a high inflammatory score, which has high granzyme K CD8 T cells, have a lower survival probability compared to those who have lower granzyme K. And so we don't understand what these cells are doing, but they're doing something. And there have been a whole bunch of other papers since that time connecting granzyme K with tumors, with infections, with other autoimmune diseases. So is granzyme K itself important, or is it just a marker of a cell subset? We would argue that the molecule itself is important, because when we take this back to the lab and we knock this gene out of a mouse and then give that mouse an experimental version of rheumatoid arthritis, this is called antigen-induced arthritis, we see that wild-type mice that have normal granzyme K have a lot of inflammation. But if the mice lack granzyme K, shown here in red, there's reduced wrist swelling and reduced inflammation. So granzyme K, the molecule, is doing something. And so people in my lab for the last several years, we've been looking at what do these cells do? We don't have all day. And so I'm just summarizing, many years of work by many individuals, some shown here and some not, who have helped us determine that granzyme K, CD8 T cells can release molecules that directly act on target cells, that act on other cells in the tissue, such as fibroblasts, to release other effector molecules, and then that granzyme K can activate complement cascades, yet another inflammatory pathway. And so, in summary, management of rheumatoid arthritis, and this is just part one, remains a clinical challenge, and it's only about 50-50 or less that any individual is going to respond to a treatment. Team science is crucial for answering these big translational questions. And that team science has revealed the cell population called granzyme K CD8 T cells that we're working on to develop to determine their mechanisms in disease. And now I'll turn over the stage to Fan.
That's great. Hey everyone, thank you, Helena. That was great. I'm Fan, and hopefully Helena convinced you how cool, using big single-cell omics data and applying different tools, we're able to see some new markers to treat rheumatoid arthritis and different ways to stratify patient heterogeneity. So, so not only our labs and our, research teams, actually many groups are actively using single-cell approach. We believe that could transform the rheumatology research. So Nature Rheumatology published many papers to summarize what could be the novel findings and the targets to carry on to really influence the patient care. So from my perspective, big data doesn't mean big discovery, right? So a lot of work to be done to transform and convert the data, the knowledge, and to the by side. So I believe, and our team has been actively working on developing different AI computational tools to find what's the key markers and the targets and the pathways. So we believe an AI-powered system immunology approach should be key to turn the massive data into actionable discovery. So Helena has shown very beautiful work of our collaboration with other teams to look at the tissue biology, tissue heterogeneity using single-cell omics. So now I'm going to show you another project our group have been doing, also as a team science, how to prevent RA, how to predict disease on site, right? It's similar, but as you hear, what I'm going to show you is totally different. So as a whole team in the division of rheumatology, so we have been working on this, building different cohorts, generating a lot of rich samples. And our hypothesis is we believe there's at-risk phase. We call the preclinical phase, before the clinical symptom. And if we could identify markers during this preclinical phase, so that markers could build based on, could be built as a target to prevent, RA onset. So that would be great to early detect rheumatoid arthritis and stop the, disease progression early. So there are a lot of hypotheses and focus underlying this research question. For example, looking at the genetic diversity, or there could be mucosal source, heterogeneity, or systemic autoimmune, immune regulators in the blood. So our focus is to see if we're able to identify certain immune signatures or pathways that drive the preclinical phase. And given this, we have completed 2 studies using large single-cell omics to study RA prediction. So the number one is also a team science. We published at JCI last year, and we developed different single-cell omics tools and methodology to identify unique marker for at-risk stage of RA. And number 2 on the right side is more using a longitudinal clinical trial, actually collaborating closely with rheumatology division here. You'll hear the 2 story and how we generate hypotheses, how we tested using different clinical and preclinical trials to validate our hypothesis and target. Okay, so for first study we did is, you can see we have 3 groups. One is established RA, the other is at-risk RA, the third is control. So control CCP negative, like Helena presented, the ACPA autoantibody negative. And for at-risk RA, we identified, use 2 parameters. One is ACPA positivity. The second is family FDR, first degree relative, because RA is also genetic disease, right? And interestingly, luckily, we have the paired mass cytometry and CySeq to measure the same individuals across these 3 populations. So that enabled us to ask the question, what are unique signatures and immune pathways in the at-risk stage compared to control or compared to RA group? And we can cross-validate using both single-cell technologies. I'll just skip a lot of technical steps, but just show you an interesting thing, a table somewhere, the T-cell focused results. Clearly, ACPA status is the main driver to separate cellular heterogeneity. But when we compare each subgroup through multivariate analysis, we found 3 populations. One is the TPH cells, T peripheral hyper cells, and also CCR2 T cells and a subset of CD8 T cell. All 3 of them are expanded in the at-risk stage compared to, healthy control. So that made us very excited. If we can look further and dive into the data to see the unique signature among these T cell subsets, we may figure out a pathway for prediction, right? So especially one population we're interested in is this T1 cluster, CCR2 positive T cells, is uniquely expanded in the ACPA positive at-risk subgroup. And what's going on with this? So thanks to the single-cell RNA-seq, you can see the T cell map on the left. But when we look at the data, we see a lot of actually signature target on the Th17 phenotype, like CCR2, IL-23R, and also Th22 phenotype, expressing a lot of IL-22 producing cells. They're all highly expressed in this CCR2 T cell population. And it's not just like 5 or 10 markers. When we look at literature and generate a score and and ask a question, which cells highly express these 2 phenotypes? That's very neat. The data told us both the Th22 and the Th17 score are uniquely, pointing to the CCR2 T cell population, and the same cells are expanded in the at-risk group compared to control. So that further confirm our, hypothesis. These two T cell phenotype or related signature could be very interesting to follow up in a more, comprehensive mechanism study. Just briefly summarize our hypothesis. So we found that two axes could build a bridge between tissue blood, as you can see we summarized here on the left. So through the CCR2 positive Th17 cells, so those cells could be recruited to the mucosal tissue because, mucosal tissue normally produce a lot of IL-22. And another pathway or axis is on the right. So through the stimulation of IL-23, and this population also express CCR2 and other, inflammatory cytokines like TNF and IL-22, and these cells could be recruited into the inflammatory joint through the CCL2 and CCR2 interaction because fibroblasts highly express the CCL2 ligand. So we are actively validating these two pathways to understand if that's true or not for next steps. Okay, this is a summary of our #1 study, and #2 is more recently, and we're very excited about. So indeed, we built a collaboration with Kevin Dean, who is the director of the preclinical RA of RA trial at Colorado University. And from this clinical trial, we're able to pull out the samples longitudinally and generate a multimodal single cell, and to answer the question, any markers drive disease progression. So in general, as you can see, we have 2 time points. For V0, we call it baseline, and we monitor, the individuals by time, and we have second time point, which is visit 1, either, development of clinical RA or still no inflammatory arthritis. And among all these individuals, we perform not just single cell RNA-seq and proteomics. You can see there are 5 or 6 multimodals because we want to understand the mechanism from different molecular, proteomics, all the different biological pathways. So also including single cell TCR, T cell receptor, BCR. Aseptic is a new technology can target both ATAC and the protein surface markers, and also serum proteomics, which is the best to identify biomarkers, right, and the genotyping. So by integrating all of them using our lab developed tools to try to understand what are the real predictor and the markers for RA prediction. So that's the goal. All right, this is just a brief summary of the time-matched sampling because I think this can give you a more visually graph. You see the green dots as the initial point, and then the purple are clinical RA, right? For mass cytometry, we have more samples than the other single cell technology because mass cytometry is cheaper. And also they are pretty well developed, established immune surface proteins, design in this study. Oh, sorry, I forgot to mention. So this is under review. If you want to read more details regarding design and also analysis, you can check out our medRxiv. So we did a lot of analysis and method development, but I still want to start from the T cell results because we see this is so interesting. Again, this time it enabled us to build a joint T cell cluster map, as you can see here, and then you can query both transcriptomic signature and the proteomic signature. And through different statistical analysis and machine learning analysis and control all the confounders, including the batch effects, age, sex, cell number difference, and we see a striking signal is the TPH cells are expanding in the RA converters before onset of clinical RA. Well, it's exciting, good to see this again, right? I showed this, we found this in the study 1, and we see the same thing in study 2. And this is longitudinal, clinical design. We confirm the prediction of this TPH. And beyond this, we also see actually 2 subset of CD8 T cells. One could be the population Helena presented, right? The Granzyme K population, or the other is the Granzyme B population. So before even we know why, but we see very clear statistics. These 2 populations are expanding in the converter before onset of clinical RA. So that means what? Oh, okay, so maybe these cells through some circulating molecules already exist in those individuals before the clinical symptom. So we're excited about this, right? As we see this expanded T cell population, we ask the question, are any of the T cell could be autoreactive, of the autoimmune disease. Then by analyzing the single cell TCR, the TCR data for the same individuals, and we're able to ask a detailed question, right? We can calculate the CD4 TCR CDR3 risk score using a predefined, coefficient value for each CDR3 amino acid sequence. If any CD4 TCR sequence is associated with RA. And interestingly, skip many steps because it's a lot of analysis between genetics and single cell immunophenotype. We just want to bring you attention about actually we do indeed see this TCR risk score, is expanded in converter than non-converter in the initial baseline point. So that indicates an expansion of potentially autoreactive T cell leading up to RA onset. So we're going forward, and did all the complete analysis for the B cell, myeloid cell, NK cell, everything. But for the time, I will not show all of them, but I'll transit to a more clinical question-driven problems is like summarized into two questions. One is conversion of RA in future predictable at baseline? The second is, are predictors associated with time or to onset of RA in converters? So we want to use this data, answer this question. You may think about, this is a small clinical cohort, not a lot of longitudinal time points, but this is the best we can do. And based on our knowledge, so far we have for the longitudinal trial in the United States to address this question. We have the best cohort, best data. So here to answer the first question, is conversion of RA future predictable at baseline? You can see we did 3 simple analyses through machine learning, classification, prediction model building. If we're only using the clinical predictor, like ACPA positivity, rheumatoid factor, age, and so on, we reach like a 0.63 for the AUC, not good, right? And when we use the cellular predictors, like what I just showed you, like TPH cells, some K cell, and other T cell, we have pretty better, like 0.73 with AUC. And eventually, if we combine both clinical predictor with cellular predictor, we have a better one is 0.76. So that gives us some confidence, by combining the new findings from our single cell analysis with clinical parameter, we could have a chance to improve the, prediction power. And again, we did all the survival analysis, and to answer another question, if this is associated with time and how soon, right? So we're able to show, the individual with higher percentage of TPH cells associated with like shorter time of onset of clinical RA, and similar of higher percentage of CCP3 titer that individual will convert to RA earlier. So similarly, we generate a, decision tree to show what are all the contributions of potential immune populations and predictors could contribute to the RA development. So far, I will show you this study, how we use single cell omics and approaches, right, to answer this disease prediction question. But this is not the end. And also computational biology, we're always interested in how we model data, how we max the power, right? How to model longitudinal single cell dynamics is a good question in this field, not just for RA. For many clinical disease, we require, clinically, longitudinal modeling. But so far in this field, not a lot of longitudinal modeling tools available because most of single cell, they are cross-sectional. They don't have the follow-up time points, right? So we're trying to address this question in our lab. The second question, I'm showing a lot of interesting things, but are we able to identify promising targets for disease prediction? We still require a lot of next step, right? Follow-up studies. Can we use computational ideas to really boost this, to prioritize target? That's another interest in the lab. I'm not going to show detail, but really showing 2 or 3 papers developed by our, postdoc and the students. So number 1 tool is called IC laser, is in review. So this is a new tool, really, identify the targets underlying longitudinal disease progression. But because for longitudinal data, it's pretty hard to analyze. There's a lot of missing samples that repeat the same measurements for the same individual, right? You have to control different tempo dynamic, technology dynamic, clinical phenotype difference, a lot of things. So on multiple time points. So this tool allows you to, really identify what are the drivers, track with time-dependent clinical outcome change. All right, so we tested on many different diseases, not just RA and other autoimmune disease in this paper. And the second tool we developed is actually explainable AI tool called Self-X. Because we know that actually using AI in genomics data or large transcriptomic data is a little bit hard, a lot of ongoing work because a lot of sometimes it's hard to interpret and hard to diagnose why this works, why this doesn't work. It's like a black box. Then we believe explainable AI is the next step we want to do. So Jade is another student in the lab developed this cool tool. And what we did is actually we really quantify and actually assign a score to each individual cell regarding their contribution to clinical outcome. So not just tell you odds ratio or p-value of cluster cells, but individual cell level, how important this cell, this phenotype, and regarding their neighbors contribute to some clinical outcomes. So why we do this? Because single cell is so heterogeneous. It's not the perfect technology, also high dropouts. You don't know what's going on. Some detections are missing, right? So this can help us understand the diversity and really look at the per cell level at once, not just a group of things. One unique application of Self-X is we can even predict the cells with sex and disease interaction effect. Why? Because autoimmune disease are mostly female predominant, and it'd be good to really see what's the difference between female and male regarding their disease severity, and how to test that is still pretty understudied in the single cell field. And we demonstrated that Self-X works in this simulation design. As you can see, we simulated different cluster of cells regarding their contribution to female sex difference, and especially just quickly in cluster I and J and AB. And through Self-X, we're able to predict the cells we simulated. Actually, B and J, they are labeled as right. That indicate those cells are really expanding in female predominant disease. And we're able to decipher the difference and link the clinical difference to the single cell dynamic difference. So we're excited to use this to other and more interesting autoimmune disease, and that'll be very cool. Also, a quick summary for this second part of our presentation. So indeed, we identify some very interesting early immune activations in the T cells, but there are also something in the B cell, and I don't have time to present, and the monocytes. And using different T cell technology, and we can identify like autoreactive T cell expansion. And we also put a lot of efforts to build like disease prediction models. And as a last point, I just want to say, longitudinal modeling with interpretive AI could translate complex data into actionable prediction marker through team science with different department and the collaborators. And I'll move to the last part. Helena and I would like to share with everyone in the next 3 to 5 minutes. So I show you for disease prediction, for predictive marker, let's go back to the tissue. And for novel treatment target, Helena has shown you a very beautiful single cell map. Many targets could be hidden there, we don't know. And we believe another single cell technology, more recently advanced technology, could help us identify the disease-driven markers, which is spatial transcriptomics. So why? I think this is not new anymore, but still pretty hard to analyze, because actually spatial omics, including transcriptomic proteomics, has been named as the method of the year by Nature Method for several times, because it highlights the high resolution and the power ability to really reveal disease-driven tissue diversity. That we believe that could be the approach when we apply it on the inflamed RA synovial tissue. And as I mentioned, right, the data is diverse and multimodal, right? You have the cell level, gene level, protein level, and the spatial level. So how you put all this information into a shared low dimension embedding and understand your data, that's our team focus on. So to make it happen, we developed many spatial omics AI methods, some published, some still ongoing to be submitted. So including 2 methods still and STEAM, and another 2 methods to be submitted soon. Just one slide for one method what it does, right? So for still, actually, as you can see, we're able to reveal the spatially informed cluster in single cell spatial resolution data, which is termed as DNAM data. You can see different low dimension embedding character, different spatial variation in the very complex brain. And it's also very scalable and fast. computationally, you can just run 10 minutes to get this beautiful picture. And another tool is called the STEAM, because one question in this field bothers a lot of biology and also computational scientists is how you are confident to claim what you identified is truth, right? Are you sure the cluster identified consistent and robust across all the spatial tissue sections, different scenarios? So we propose this framework to evaluate the robustness and consistency of the spatial-driven clusters. So you can use that to evaluate their results and improve their results. So with those tools available and our biological hypothesis target available, so Helena and I will building team science or indeed collaborating with the HIMSR, right, the single cell core to generate a lot of beautiful data.
Yeah, and so Fan and I, we've been working together now for it's almost 10 years, first in Boston and then after coming here. And what I love about our collaboration is that we sort of, in a two-person system, are able to span, everything from sort of like clinic to bench to computational and then going back and forth. And so you heard that clinical case at the beginning and some bench science, and then you heard the computational algorithms that Fan's lab are like churning out to help answer these really important questions and connect these data sets together. And so now I'm going to tell you about, in just a minute or two here, about this collaboration that we're starting out. We just got our first batch of data, like literally about 10 days ago, that we're super excited about. And so just to sort of take a step back and put this back into context. So again, we have those tiny little synovial biopsies. If we looked at each one of them under a microscope, it would be, there's architecture in there. There's lining and sublining and lymphoid aggregates and so forth. I showed you that histology earlier where there are sometimes lots of T and B cells and sometimes not, and they form these structures. But when we do that big study like we did a few years ago, we like literally put them in a blender. We put them in a blender, we create a cell smoothie, and then we look at the cells in the smoothie and we can classify them. Like this is a graph of the different categories of cells, but we have no idea who's close to whom. And so we're stuck with a cell smoothie. It's delicious, it's like nutritious and helpful, but it's not what we started with, right? Like we don't know if the grapes are talking to the melons or if they're talking to the blueberries, and that's important for knowing how to target them. So spatial omics, so spatial transcriptomics, spatial proteomics allows us to keep that fruit tray intact, to figure out if the cantaloupe is friends with the strawberries or whether it's more like interested in talking to the pineapple. Because these communication molecules, that's actually been the most successful way to treat autoimmune disease, right? TNF blockers, IL-6 blockers, et cetera. And so these are some data that we've already used to push our project forward. So this is looking at a piece of synovial tissue, one of those teeny, tiny fragments. This is probably about 100,000 cells that we see on here. Each cell is like a teeny, tiny little dot that's probably about, like 1/40th the size of the red laser pointer here. Yellow marks the granzyme K cells. And what we find is that they're located in the same place as cells that express CD68, which is a macrophage or myeloid cell marker. They do not happen in the same places as CD19, which is a B cell marker. This tells us that granzyme K CD8 T cells are located next to and communicating with myeloid cells, not B cells. So when we're trying to model this in the lab, et cetera, we need to focus on myeloid cells. And so that's one way we're using this simplistically, but we're also hoping to sort of like bring this into this like really complex web of bench science, clinical information, computational methods, et cetera. And like we're really excited about how the Jonsson lab and the Zhang labs bring together all these different skill sets to push this forward in important ways, where we integrate the pathology, the biology, these different algorithms here. Like we can use published data sets that are big and very well annotated. We can use our own tiny little samples that were collected here from the pathology shared resource, from the Interdisciplinary Joint Biology Consortium, this collaboration between rheumatology and orthopedics that's collecting synovial tissue, and gather it all together so that we have computational, bench science, and then clinical applications all working together. And this is where this collaborative project lives together. So we have Nancy and Sam in the Zhang lab, Kaylee and Connor in my lab, working together to bring this really complex data set that's at the forefront of translational science developing the methods to integrate the protein staining that we're doing with the omics data that we're generating to help, learn something new about rheumatoid arthritis that then we can hopefully take back to clinic and eventually generate treatments. And so these are the members of the Jonsson lab. Many of them are here today that have really been working on this from multiple different angles all at once. There are also many other people at the University of Colorado that I want to thank, Fan, obviously, and her whole lab, Mike Hollers, Larry Moreland, who's been really instrumental in initiating the synovial tissue biorepository that we have here, without which none of this would be possible. We have people in the clinic who do biopsies, Melissa Griffith, Mila Kasianek, and then again, this interdisciplinary joint biology program that helps to coordinate getting these samples because it's not easy, and the rheumatology clinic staff. And then we also have members of the lab where I did my postdoc in Boston and my funding sources. And then Fan has her acknowledgments here. I think she talked about people along the way. And with that, we are happy to take any questions.
Thank you.
Great, thank you. I'm going to go ahead and open it up for any initial questions that people have. While people are thinking, it was really fascinating to me how the cellular composition is so different in different people. And you mentioned how it changes over time in the setting of treatment. Is that due to treatment, or does it change over time in everybody, or is this just like sampling error? Do we know anything more about how that changes in people over time?
Yeah, I think right now we don't know if it changes only with treatment or if it can only change with time, because the only setting in which we have these repeat biopsies were in the setting of a clinical trial where people started a medication between those two biopsies. And so in the UK, they're doing this a lot with the National Healthcare Service. This particular study were people who started on methotrexate, the medication I mentioned earlier. If they didn't respond to methotrexate, the UK, like National Health Service, allows them to either start rituximab, a B-cell depleter, or tocilizumab, which is an IL-6 receptor blocker. And so they kind of did this informal randomization and then followed people. And then so everyone received a treatment. The changes we saw comparing, like in the people who got rituximab, the B-cell depletion versus the IL-6 blocker were not exactly the same. We saw like the T and B cells go away more in the people who received the B-cell depletion compared with the tocilizumab, the IL-6 blocker. But that study, like it only had about, there were about 40 people who got these two sets of biopsies, and it's not enough. Like humans are too diverse. 40 people in a study is not enough. And so we're hoping to encourage people who do clinical trials to include synovial tissue biopsies as part of that, so we can build on this number. The nice thing with these new technologies is that it doesn't have to be in one clinical trial. Like we can take those 40 samples and then add 40 samples from a different clinical trial. As long as they use RNA-seq of some sort, we can integrate those two samples together and analyze them together, and then we have an N of 80 instead of 40. And that happens like more and more and more. And so we're hoping to see that bridge between, the computational bench, clinical trial, clinical care sort of connect better, because that's going to help us make progress faster.
Great, thank you. Yes.
Rusty.
All right, very great presentation. I guess looking at how you see the immunopathology through the computational sciences and in what you can recapitulate at the bench, what does this tell us about the course of disease from maybe preclinical to clinical to post-treatment, and how that may change our perspectives if we're taking care or trying to prevent disease versus what we're going to use to treat disease, or maybe treat more later onset, or not late onset, but more chronic disease?
Mm-hmm. And so I think, feel free to jump in. I think where Fan's project is really amazing with this whole progression to disease is identifying who will progress. And it identifies both biomarkers, because again, if we had a blood test where we could maybe test that woman, that I talked about earlier who got rheumatoid arthritis right after pregnancy, if there had been a test a couple years before that would have said you have a really high risk, then maybe we could have done something different there. But what I think that Fan's project also does is give us leads about cell populations that might also be actually conveying that risk, and so these CCR2 positive CD4 T cells, that are Th22 and Th17 phenotype and stuff, give us ideas about mechanisms. So for example, with that, IL-17 blocking therapies are really useful in psoriasis and psoriatic arthritis, but have not worked for established rheumatoid arthritis. But maybe they would work for people who are at risk, right? So if we gave my patient, the pregnant patient, the one who'd just given birth earlier, like if she'd gotten an IL-17 blocker maybe 2 years earlier when her blood test showed high risk, maybe that could have prevented the rheumatoid arthritis to begin with. And so treatments that already exist and that work well in other diseases could very easily be adopted for a different purpose. And we might need different treatments to prevent progression compared to treating disease.
I totally agree. I think the timing is very important. As Chris you mentioned, is like before preclinical, right, and early disease, and then late or before after treatment. And we see actually those signatures they change differently across the whole disease progression time. And one thing we know that actually because we're limited, right? One lab or one team can only generate these small data sets. And one thing we believe the data science, that's the power, is we're trying to develop integrative methods to pull all the related data together from the public domain. Because your lab generated after treatment, before treatment, and my lab generated like a before clinical onset data, right? But I know there are different cohort difference, right? And patient, population difference. But how we use all this information to generate big data, right? And maximize the power to dig into data. What's the most, effective treatment for which stage of the disease time course? I think that's the next step because we need the data, but data is not perfect, right? Sometimes garbage in, garbage out, right? And we need to learn and make the generalizable marker. We need to test on multiple cohort, how to do the cross cohort integration, how we validate data from Colorado cohort, right? Boston cohort or California cohort, right? That's what we're doing actually, now for method development. But we'll also feel really lucky to have this Colorado cohort data, which is the first one, we can generate hypothesis and to be tested in other cohorts.
Wonderful. Well, thank you so much. We're at the end of time, but we appreciate you coming.